Hi students, last class we discussed about the sexual reproduction. In that case we discussed about the different parts of the flower and essential parts and non-essential parts. Within essential parts we discussed about the androecium and the gynoecium, right? So in the last class we discussed about the stamens, regarding to the stamens. Within that case anther is the very important part, right? In that case we discussed about different layers which are seen in anther, cross section of anther, right? And coming to the next class, means coming to this class, we are discussing about the three-dimensional cut of anther. See, this is one of the anther structure. In that case, right? See, this is one of the anther structure. In certain cases, okay, anthers here. This is one type of anther, right? In certain cases, the anthers are attached like this. So if we cut the anther, so this is the view. This is the exact view. So here there is a different parts are present within the anther. Different parts are present. So exactly this view, three-dimensional cut section of anther. This part is the anther. This part is the filament. This part is the anther. This part is the, right, up to this part, this is the anther, right? So this is one of the anther part. Within that case we discussed the last class also, outermost part, epidermis is there, endothelium is there, right? There is a middle layers are there, and next one there is a tapetum tissues are there, tapetum layers are there, right? Sporogenous tissue, these are nothing but pollen sacs. See, epidermis is there, right? And next one, yeah, endothelium is there, middle layers are there, right? The central tissue is the very important one. The central tissue, this is the one of the central tissue. This is one of the central tissue. This is one of the central tissue. So this tissue, okay, this is exactly, okay, called as sporogenous tissue. The tissue is called as sporogenous tissue. Sir, why this is sporogenous tissue? See, the sporogenous tissue involving the production of pollen grains. The sporogenous tissue involving the production of pollen grains. See, what I am telling, whatever that microspore mother cells are there, before to, okay, these pollen grains, there is a microspore mother cells are there. The microspore mother cells undergoes meiosis process, okay, which are produces the pollen grains. So the microspore mother cells, the tissue is called as sporogenous tissue. That sporogenous tissue is nothing but spore mother cells. That spore mother cells undergoes meiosis process, produces the pollen grains, right? So this is one of the cut part of the anther. See, here these parts are very important. The central parts are very important. These are called as pollen sacs. So within the anther, this is one of the bilobed anther. How many pollen sacs are present? This is the first pollen sac. This is the second pollen sac. This is the third pollen sac. This is the fourth pollen sac. So in bilobed anther, 4 pollen sacs are present, and all these 4 pollen sacs contains spore mother tissue or sporogenous tissue or microspore mother cells, which undergoes meiosis process, produces the pollen grains. Then these pollen grains are released out. See, there is an specialized grid-like structure or specialized groove-like structure is produced within the anther. Specialized groove-like structure is present. Okay, that groove especially made up of with special cells. The special cells are called as stomium. The stomium cells, okay, especially this groove, continuously this groove contains the stomium cells. Stomium cells are involved in dehiscence process. Dehiscence is nothing but breakdown, open. The cells, whatever the cells are there, the cells are broken down, right? Once the cells are broken down, okay, the pollen grains are released out. The cells are, okay, broken down, okay, the cells are broken down, pollen grains are released out. So here the cells broken down process is called as, cells breakdown process is called as dehiscence. cells breakdown process is called as dehiscence, right? So once the dehiscence takes place, the pollen grains are released out. The pollen grains are released out, right? So that is about the three-dimensional cut section of anther, right? Remember, see, within that three-dimensional cut section of anther contains 4 pollen sacs. See, particularly this one part, particularly this part is called as sporangium. This is the one sporangium. This is called as sporangium. This particular part is called as sporangium. This particular part is called as sporangium. So, especially this pollen sac region, okay, that one is called as sporangium. Now we discuss about the structure of sporangium. Now we discuss about the structure of sporangium, right? So this is about the cut section of anther. Cut section of anther, right? Coming to the next structure is the structure of microsporangia, right? We discussed about the anther and different parts of the anther and different types of the anthers, monothecous anther and the dithecous anther. And coming to the next one is, within anther part there is an microsporangias are there. Microsporangias which are responsible to produce the pollen grains. Microsporangias are responsible to produce the pollen grains. Sir, what are the layers or what are the tissues are present in the anther? What are the layers or tissues are present in the anther? See, any structure, there is an outermost structure is there. The outermost structure is called as, within the plants, right? This is called as epidermis. We know most of the living organisms contains outermost layers are called as epidermis, right? Here also there is an epidermis is there. The blue color lines are indicates here epidermis, right? At some regions, see, these are the special cells or the special tissue, right? See, these cells are called as stomium cells. These cells are called as stomium cells. See, within the microsporangia, at some regions, very thin cell wall contained cells are present. These cells are called as stomium cells, right? See, these stomium cells are involved in the dehiscence process. The stomium cells involved in the dehiscence process. What is dehiscence? What is dehiscence? We know one of the young anther is there. For suppose this is one of the young anther. The young anther contains the tissues are there, right? And the young anther developed into the mature anther. Within mature anther, inside the anther, there is a many number of pollen grains are present. This is the one of the mature anther. This is one of the mature anther, right? That mature anther contains the pollen grains. See how the pollen grains are released. How the pollen grains are released. See, by the degradation of anther walls. By the degradation of anther walls. See, if the anther walls are broken down, if the anther walls are broken down, the pollen grains are released out. The pollen grains are released out. So, breakdown of anther walls, right? The process is called as dehiscence. The process is called as dehiscence. So, the stomium cells are very thin cell wall contain cells which are involved in the dehiscence process. That is nothing but breakdown process, right? So, the breakdown from outside started from the stomium cells. The breakdown of cells started from the stomium cells, especially from outside, right? And see, I already discussed about epidermis, the blue color. Okay, the blue color cells are nothing but epidermis, right? See, first of all, we'll discuss about the total parts. After that, we'll discuss about the one by one, right? See, epidermis. This is one of the uniseriated structure or single cell layered structure. Single cell layered structure, right? See, epidermis majorly involved in the protection. The very first function is the protection, right? After that protection, the next function is the dehiscence. The very first function is the protection and next function is the dehiscence. So here the epidermis involved in two functions. One is the protection, other one is the dehiscence. Protection is nothing but, we know our skin, okay, what are that, okay, we have that skin, This is nothing but one of the epidermis, outermost part, right? The skin major function is protects the inner organs, In microsporangia also, whatever that epidermis is there, epidermis involved in the protection, followed by the dehiscence, right? And inside to the epidermis there is a very special tissues there, right? The special tissue is called as endothelium. Inside to the epidermis, very special tissue is there. That special tissue is called as endothelium, right? Sir, why endothelium is the special tissue? Because it contains the callous strips. See the callous strips which are made up of with the alpha cellulose, right? The callous strips especially made up of with the alpha cellulose, right? So presence of alpha cellulose, the callous strips formation takes place. Then what is the function of endothelium or the callous strips? See, simple, both these are involved in the same kind of the function. See, the endothelium showing the hygroscopic movements. The endothelium majorly involved in hygroscopic movements. Hygroscopic movements is nothing but absorption of water from the atmosphere. Atmosphere contains whatever that moisture content is there, that moisture content directly absorbed by certain types of special tissues, especially these are also seen in anther. Within the anther, right, the special tissue is called as endothelium. Endothelium shows that hygroscopic movement or hygroscopic absorption. Exactly here. Okay, the better word is here, not exact movement here. The better word is the absorption, right? Hygroscopic absorption, right? So here water is absorbed from the atmosphere, right? That is about the endothelium, right? How the endothelium have the capacity to absorb the water from the atmosphere because of the cellulose. We know about that cellulose, especially that is seen in plant cell walls also. But cell walls contain cellulose percentage is very low. It's not have the capacity to absorb the water from the atmosphere. Thus, cell walls contain cellulose, not have that much of capacity. But these are the pure cellulose bands, okay, which have the capacity to absorb the water from the atmosphere, right? So, see the hygroscopic absorption, that is nothing but absorption of water from the atmosphere, right? So, then what is the use of hygroscopic absorption? See, this one is also supporting for the dehiscence. See, all these are involved in dehiscence process. See, coming to the next one, middle layers. See, inside to the endothelium, there is a 2 to 4 layers are there, which are called as middle layers. Middle layers are involved in the transportation of nutrients and transportation of water. Right? That is about the middle layers, right? And coming to the next one, tapetum. This is a very quite different tissue from remaining all anther tissues. See, this is the very important tissue for the anther. Or microsporangia, because the tapetum involved in the nourishment. Of microspore mother cells. Whatever that tapetum cells are there, the tapetum tissue is the very special tissue compared with the remaining all tissues, because the structure also different. Okay, we will discuss later, right, one by one regarding to these parts. So the tapetum is a very special tissue involved in the nourishment of microspore mother cells. The last center one is this is the center part, right? See, the center one is the microspore mother cells. The center black color cells are there here. The black color cells are indicates here microspore mother cells. So what is the what is the function of microspore mother cell? Simple. The microspore mother cells which undergoes meiosis process. which are involved in the production of pollen grains, pollen grains. So what I am telling here, whatever that the center cells are there, the center cells within that microsporangia, okay, these are the very important tissue and these are the fertile tissue. Fertile tissue means, okay, the cells have the capacity to produce the pollen grains. The cells have the capacity to produce the pollen grains. So the central tissue is the very important tissue. These cells are called as microspore mother cells, which are undergoes meiosis process. We know within meiosis process, one cell, right, one parental cell produces the 4 daughter cells. One parental cell produces the 4 daughter cells. Whatever that 2 end state cells are there, the 2 end state cells produces the haploid state cells or end state cells, right? See, these are nothing but pollen grains. These are nothing but the pollen grains here. Here within microsporangia, the microspore mother cells, okay, the cells undergoes meiosis, which are produces the pollen grains, right? So that is about the, okay, the basic discussion regarding to that structure of microsporangia. One by one we will discuss about the each part of the microsporangia, right? See, this is a very important question. Okay, this is a very important topic. Okay, in the state examination, this is a very important IPA question for 8 mark question. This is one of the 8 mark question, right? In the NEET examination point of view, this is the one of the important area. See, from this area, most of the bits are given. Right? So we'll discuss about one by one part of the microsporangia, right? See, the very first cells here, I discussed about the stomium cells, right? These are the stomium cells. See, these are very special cells. And which are having thin cell wall. Right? Thin cell wall contains cells. Okay. Thin cell wall contains cells. Once these cells losses the water, right, which are involved in the dehiscence process. See, dehiscence is nothing but breakdown of anther walls to release the pollen grains. Right? That is about the stomium cells. And coming to the next one is the epidermis. And the very next part is the epidermis. Right. See, the next part is the epidermis. Epidermis is the single layered structure. Single cell layered structure. Right. So epidermis is made up of with single layered cells. This one is majorly involved in the protection, protects the inner tissues. Right. Other one is the dehiscence. Right? So these are the common points, but the major difference here, okay, when we are discussing about the endothelium, when we are discussing about the middle layers, when we are discussing about the tapetum, these points are different. Generally, these 2 points are similar. Okay, what I discussed earlier, okay, the same points also I repeated here. But we have to write all these points in the running notes. Right? All these points are very important. Epidermis is made up of it. Okay, epidermis is a multi-layered structure. Epidermis is a 2-layered structure. Epidermis is a 3-layered structure. Epidermis is a single-layered structure. So this is the way of bits asking in the NEET examination, right? So that's why we have to prepare. We have to remember every line of the NCERT, okay, in a proper way. Then we analyze easily. We analyze the bits which are given in the NEET examination, right? So that is about stomium and the epidermis. And coming to the next one is the endothelium. I said the endothelium is the very special tissue. Right. Endothelium is the very special tissue, the tissue present inside to the epidermis, right? See, the cell shape is also quite different. See, normal shape, okay, normally maybe the round shape, maybe the barrel shape, maybe the oval shape, but these cells are not like that. Okay, the shape is quite different. The shape is quite different, right? See, these are maintained very special kind of the structure. The endothelium cells are maintained the very special kind of the structure, and the center of the cell, okay, this one is also single layered structure. Endothelium also single layered structure, right? The center of the cell, center of the endothelium cells, okay, there is in bands are present. Bands are there. See, these bands are called as callus strips, which are especially made up of with the alpha cellulose, So the alpha cellulose, whatever that callus bands are there. Okay, see, there is in callus bands are there. The callus bands which are especially made up of with the alpha cellulose, right? That alpha cellulose is responsible for the absorption of water. The alpha cellulose bands are responsible for the absorption of water from atmosphere. That alpha cellulose is responsible for the absorption of water from atmosphere. So this step is called as hygroscopic movement or hygroscopic absorption. Hygroscopic absorption. So, see, the special tissue have the capacity to absorb the water from the atmosphere. Sir, if these cells absorb the water from the atmosphere, what happens? Okay, see, in the mature anther, see the cells, whatever that the endothelium cells are there, the endothelium cells are dehydrated. Once the endothelium cells loses the water, then the cells are bursted. That is nothing but the cells are undergoes breakdown process. Once the cells are undergoes the breakdown process, that is nothing but dehiscence. Right? So here what I am telling, the simple concept, what I am telling here, whatever that cells, endothelium cells are there, normally which absorbs the water. If the water is there, the cell structure is safe. Okay, the cell structure is safe, means the cells are maintain the proper shape. Okay, the cells are alive. Once the cells loses the water, okay, even in environment also there is no availability of the water, then these cells are degraded. If these cells are degraded, then okay, the step is nothing but dehiscence. Simple. The endothelium, very special tissue, having that callus strips which are made up of with alpha cellulose. Cellulose is responsible for the absorption of water from atmosphere, especially the function carried out in young anther, in mature anther. Okay, once the cells loses the water, right, the cells are broken down. That is nothing but dehiscence process. That is about the endothelium. Coming to the next one, middle layers. Middle layers, right? Inside to the endothelium there is 2 to 4 layers are there. 2 to 4 layers are there, right? Which are nothing but middle layers. In some anther maybe 2, in some anther maybe 4, right? In some anther maybe 3. Okay, the layers is differ from one anther to the one species anther to the another species anther, right? Maybe in hibiscus, okay, maybe 3 are maintained. Maybe capsula, maybe 4 are maintained, right? So here what I am telling, the middle layers, the range is 2 to 4 layers. Sir, what is the function of the middle layer? What is the function of the middle layer? Or what are the functions are performed by the middle layers? Simple. The middle layers are involved in absorption of water, and nutrients. Absorption of water and nutrients. See, the middle layers involved in the absorption of water and nutrients. So after absorption, okay, what about the nutrients? Or what will do the nutrients? What will do the nutrients by the middle layers? See, actually, the middle layers are supporting for the absorption of water and nutrients. After the absorption process, the middle layers again involve in the supply of nutrients to tapetum cells. Supply of nutrients to tapetum cells. See, simple. See, whatever that middle layers are there, the middle layers involved in the absorption of water and minerals, right? After that absorption process, these water molecules and the minerals or the nutrients supplies to the tapetum cells. Right? That is about the middle layers. Remember here, number of middle layers are very important. Right? That is about the middle layers. Coming to the next one, very important topic is the tapetum. Coming to the next one is the tapetum, right? Within tapetum, okay, these are the tapetum tissue or the tapetum cells. The very first point is the very strange point. See, these are binucleated or multinucleated. These are binucleated cells or multinucleated cells. These are binucleated cells or multinucleated cells. Generally, we know about the normal cells. I am discussing about the normal cell. This is a normal cell. Only one nucleus is present. 99% of the cases, right? One nucleus is present. See, the condition is called as uninucleated condition. But in tapetum cells, this is one of the tapetum cell. The tapetum tissue is quite different from remaining tissues. Generally, this is the binucleated, means two nuclei are present. Generally, the tapetum cells are binucleated, right? Means two nuclei are present, right? And next one is the so in some cases multinucleated. Maybe 3 or more than 3 nuclei also present. So tapetum cells are very special tissues, which are binucleated condition or multinucleated condition, right? So that is about the first point regarding to the tapetum cells, right? See that tapetum cells, the second point is here, the tapetum cells. Regarding to the tapetum cells, the second point is storage of nutrients. Tapetum cells are involved in the storage of the food material. And next function is nourishment. Nourishment of microspore mother cells. Whatever that the tapetum here, red color layered, right? Red color layered structure is there. That tissue is called as tapetum tissue or the cells, tapetum cells, which are involved in the storage of nutrients and which are also supplies nutrients to the microspore mother cells. See the center part, the black color cells are there, which are called as microspore mother cells. That microspore mother cells are nourished by the tapetum cells. Tapetum cells, right? So that is about the tapetum cells and the nourishment process. This is one of the function. And the tapetum cells also involved in the secretion of sporopollenin. Tapetum cells also involved in the secretion of sporopollenin. Sporopollenin, right? Sir, what is sporopollenin? We didn't hear that name. Okay, before to this class. Okay, we never hear the sporopollenin word. See, we know about the pollen grain. I thought everyone heard about the pollen grain. This is the structure of the pollen grain. This is the pollen grain, right? See the pollen grain, outermost structure. This is the outermost structure, right? See the outermost structure unevenly present, means highly decorated structure. The pollen grain, outermost structure is there. The outermost structure is highly decorated structure. See that structure, okay, the highly decorated structure is made up of with the sporopollenin. Actually, the outermost structure is called as exine. The outermost part of the pollen grain, the part is called as exine. Exine is made up of with the sporopollenin. Exine is made up of with the sporopollenin, right? So that is about the exine. Okay, exine majorly made up of with the sporopollenin, right? So within pollen grain, whatever that outermost structure is present, the outermost structure is called as exine, right? The exine majorly made up of with the sporopollenin. Sporopollenin is secreted by tapetum. Sporopollenin is secreted by tapetum cells, right? So that is about some of the functions. And remember, one of the important function is tapetum cells also involved in the secretion of pollen kit. Secretion of pollen kit. What is pollen kit? We will discuss. Right. Okay, tapetum cells also involved in the secretion of pollen kit. Tapetum cells also involved in the secretion of pollen kit. What is pollen kit? See, within pollen kit, the very first one is, okay, the tapetum cells majorly secretes the oily substance. Oily substance. See, so what is the use of oily substance to the pollen grains? For suppose one of the pollen grain is there. The outermost part of the pollen grain always having one oily or gel-like substance, because okay, if we touch the pollen grains, okay, the pollen grains are attached to our hands due to oily substance. For suppose this is one of the flower and this is one of the flower, right? So here this is one of the insect. This is one of the insect, right? So the insect comes land on the flower. Okay, we are discussing about here insect pollination. How the insect pollination takes place. See, when the insect land on the flower, that time some of the pollen grains are attached to the insect. So how the pollen grains are attached to insect? Due to the oily substance. Right? So due to this oily substance, the pollen grains are attached to the insect. Then insect pollination will be takes place. So here pollen kit is supporting for the insect pollination. So that is the first point. Oily substance is the first point within pollen kit. And pollen grains exhibit the yellow color. Okay, see, whatever that pollen kit is there, that pollen kit supporting for the synthesis of certain pigments. These pigments These pigments especially gives the yellow color. These pigments gives the yellow color. Sir, what is the use of yellow color? See, whatever the pollen grains are there, if we see in most of the pollen grains, the color of the pollen grain is the yellow color. The color of the pollen grain is the yellow color. Because the yellow color is easily attract the insect. Easily attracts the majority of the insects. Honeybees also, okay, very easily recognizes the yellow color. So what I am telling here, when the pollen grains are having yellow color, okay, the yellow color easily attracts the insects. Then the pollen grains are attached to insects. The pollen grains are attached to insects. Then there is an insect pollination will be takes place. There is an insect pollination will be takes place. So here what I am telling, pollen kit, within pollen kit, oily substance is there and yellow color substances are there. These are nothing but pigments, right, which are having yellow color to attract the insects, right. So that is about the tapetum cells. See, tapetum cells especially involving nourishment of spore mother cells. Nourishment of spore mother cells. The very first point is storage of food material or storage of nutrients. Next point is, okay, nourishment of microspore mother cells. Next point is there is an sporopollen secretion is takes place. The pollen grain exine formation done by the sporopollen. Sporopollen secretion done by the tapetum cells. And next one is the pollen kit. Next one is the pollen kit. There is an oily substance and yellow color pigments. Yellow color pigments are synthesized by the pollen kit, right? So that is about the tapetum. Remember, the tapetum is very, very important. The tapetum cells are either binucleated cells or multinucleated cells. Binucleated cells are multinucleated cells, right? That is about the tapetum. So remember all of you, tapetum cells also in mature anther. In mature anther means, see, these are the tapetum cells. If the nutrients are supplied to the microspore mother cells, the tapetum cells are degraded. The tapetum cells are degraded, right? See, the middle layers are degraded, right? See, whatever the endothelium cells are there, which are degraded, right? So epidermis cells are degraded, right? In mature microsporangia, all these cells are undergoes the breakdown process, right? Then whatever that microspore mother cells are there, which are already, okay, produces the pollen grains, because in matured anther only pollen grains are present and these tissues are degraded or degenerated, right? The pollen grains are released out. The pollen grains are released out, right? So here what I am telling, the tapetum cells are also involved in dehiscence process. The tapetum cells are also involved in the dehiscence process, right? So that is about the tapetum cells or the tapetum tissue. The last one, microspore mother cells. Microspore mother cells. Yeah, microspore mother cells. These are also called meiocytes. Simple. Which cells are involved in the meiosis process? Those cells are called as meiocytes. Either in microspore mother cells, okay, either megaspore mother cells. Both are called as meiocytes. Those the cells are involved in meiosis. These cells are called as meiocytes. These cells are called as meiocytes. So here the meiocytes, the state is the 2n state, right? The meiocytes are the microspore mother cells which are undergoes meiosis process. Which are undergoes meiosis process, and these cells, okay, involved in the production of pollen grains. One microspore mother cell. This is microspore mother cell. That microspore mother cell undergoes meiosis process, produces pollen tetrad. Produces pollen tetrad. So what is pollen tetrad? See, we know in meiosis process, one cell produces the 4 daughter cells. One cell produces the 4 daughter cells. So pollen tetrad is nothing but one microspore mother cell. This is the microspore mother cell. This is covered by one of the layer. See, the layer is called as callose layer. The layer is called as callose layer, right? So already that is covered with the callose layer or callose membrane. and inside the microspore mother cells undergoes meiosis process. So 4 pollen grains are present. 4 pollen grains are present inside the callose membrane. Inside the callose membrane. So this structure is called as pollen tetrad. Pollen tetrad. Tetrad indicates 4. Right? 4 pollen grains are present inside the callose layer. So this one is called as pollen tetrad. Pollen tetrad. So the pollen grains are released 4 at a time. From the anther, 4 pollen grains are released at a time. No. See, again, the callose is the one of the chemical substance. The callose is cleaved by the callase enzyme. Callase enzyme, right? Once the callase enzyme is released, the callose layer is broken down. Then the pollen grains are free. The pollen grains are free, which are released. That is nothing but, see, these are the microspore mother cells. These are the microspore mother cells, which undergoes meiosis process. So inside, many number of pollen grains are produced. These are nothing but pollen grains. Here pollen grains, okay. Here pollen grain state is the end state. Here pollen grain state is the end state. Many number of pollen grains are released here. Once the pollen grains are produced, right? See, whatever that tissues are present here, okay. These are already degenerated because once the tapetum cells are supplies the nutrients to microspore mother cells, right? See, the tapetum cells are degenerated. Because already these are supplies the nutrients. So what is the use of nutrients to microspore mother cells? See, the microspore mother cells undergoes meiosis process. Meiosis process requires energy. That energy supplied by the nutrients. The nutrients are supplied by the tapetum. Right? So tapetum cells supplies the nutrients to microspore mother cells. That energy utilized by the microspore mother cells, which undergoes meiosis, produces the pollen grains. So here, after the nutrient supply, the tapetum cells are degenerated. The middle layers are degenerated. Right? The endothecium cells are degenerated. The anther walls are, okay, whatever that epidermis is there, epidermis is broken down. The pollen grains are released out. Right? So finally, okay, the pollen grains are released out. Right? So that is about the anther structure. Okay, within the anther, okay, this is nothing but microsporangia. So within microsporangia, these are the tissues are present. Epidermis is there, endothecium is there, middle layers are there, tapetum, right? Tapetum is very important one. After that, microspore mother cells are there, right? So that is about the structure of microsporangia. Right?